This video discusses a paper exploring the effects of first and second generation antipsychotic drugs, haloperidol and olanzapine, on neural networks in vitro. Published in Nature's Scientific Reports in 2017, the main finding of this work is that these drugs modify network density, connectivity, and spontaneous firing patterns in vitro. This supports an understanding of schizophrenia revolving around decreased power of gamma oscillations due to imbalances in inhibitory and stimulatory signaling. The scientific community's understanding of the pathophysiology of schizophrenia is relatively limited. However, most theories revolve around the imbalance of neurotransmitters, imbalances of inhibitory and excitatory signaling, and abnormal neural connectivity correlated to patients with schizophrenia. One way of investigating the pathophysiology of schizophrenia is by exploring how antipsychotic drugs alleviate psychotic symptoms associated with the disease. The significance of this work stems from the difficulty and importance of effectively treating patients with schizophrenia and related psychotic disorders. These diseases affect approximately 1% of the population and can be severely debilitating to those affected. In addition to the significant impact on quality of life for the individuals, these diseases cause a heavy financial burden on society. The main discovery and point of the paper is that first and second generation antipsychotics modify the network density, connectivity, and spontaneous firing patterns of neurons studied in vitro. The first study discussed in this work examines the inhibitory and excitatory synapse connectivity in the presence of olanzapine or haloperidol. Pre- and postsynaptic markers are tagged with fluorescent antibodies to indicate the prevalence of inhibitory or excitatory synapses. Figure 1a and 1b display the theoretical basis of fluorescently tagging these synapses. For example, blue fluorescent antibodies are used to tag aggrecan, which is an indicator of inhibitory interneurons. Similarly, PSD95 and VGlut1 indicate excitatory synapses, indicated in green and red, respectively. Finally, Gefferin and VGAT tags indicate inhibitory synapses. Fluorescence data was collected in the presence of each drug, resulting in images such as the one shown on this slide. Using the immunofluorescent technique mentioned previously, we can observe how synaptic connections change between the treated neurons. Classically, first-generation antipsychotics, such as haloperidol, are known to decrease neuronal activity, while second-generation antipsychotics, such as olanzapine, are known to increase neuronal activity. However, the data in Figure 1d shows that the effects of the drugs are more complex than the traditional understanding. We can see that olanzapine significantly increases excitatory input to excitatory neurons, while haloperidol increases inhibitory synapses to inhibitory neurons. Both effects lead to increased neural activity. However, both olanzapine and haloperidol increase excitatory input to inhibitory neurons and increases inhibitory input to excitatory neurons. This would decrease overall neuronal activity. This data suggests that haloperidol and olanzapine work in a more complex process to regulate neuronal activity instead of one only simply increasing or decreasing neuronal activity. In Figure 3, we can see an illustration of the experiment that was done to see how the antipsychotics affected neural connections at different points in neuronal development in vitro. Part A shows the multiple electrode array that neuronal culture was grown on to detect electrical activity. Each of the black dots represents an electrode on a plane that can measure electrical activity of 5 to 10 neurons firing. Part B of this figure shows the electrical activity of neurons of the entire multiple electrode array recorded over 15 minutes. Each square in Part B represents the recorded activity of an electrode in Part A. Note that each square in Part B is located at the same location as its electrode in Part A. Part B prime shows an average bursting pattern measured in a single electrode. Each spike in Part B prime represents a burst. Part B double prime shows a line graph that represents a single burst structure, which is the structure of the spikes, or bursts, seen in Part B prime. This graph demonstrates how olanzapine and haloperidol influence firing rates at different points in development. At 7 days in vitro, both drugs result in significantly lower firing rate than seen in control. By 14 days in vitro, these drugs produce firing rates similar to that of control, but with olanzapine slightly higher. And at 21 days in vitro, olanzapine produces firing rates much higher than that of the control, and haloperidol produces firing rates lower than that of the control. The second figure in this graph shows how antipsychotics affect bursting rate at different developmental points. we can see that at 7 days in vitro, olanzapine and haloperidol both resulted in mean bursting rate lower than that of the control. Then, at both 14 and 21 days in vitro, olanzapine resulted in a mean bursting rate higher than the control, and haloperidol resulted in a mean bursting rate lower than the control. Overall, this study found that both olanzapine and haloperidol have appreciable effects on the proportions of different types of synaptic connections. One difference between the effects of these two drugs was that haloperidol increased the number of inhibitory to inhibitory synapses, while olanzapine increased excitatory to excitatory synapses. In addition, both of these drugs showed significant effects on the firing rate and bursting rate of neurons. Olanzapine generally increased these values, while haloperidol generally decreased them. However, the results of the study is to indicate that these drugs' actions is more complex than this. These insights have the potential to lead to improved treatment of those who use antipsychotics. Antipsychotics are known to often result in many undesirable side effects, some of which discourage patients from continuing their medication. Studying the effects of antipsychotics on neural activity could give us insight into what causes these side effects and potentially lead to the development of better drugs that have less side effects.